So we have this population of descending neurons, and you're trying to show sort of the connectivity map from brain areas onto ventral nerve cord areas.
Exactly.
Even in this visualization, I'm wondering, like, what kind of options do we have for these connections? Like, can we do like nodes? Jeremy is an expert at visualizations and data processing and understanding large data sets. And so we're working with him to try to visualize the connectivity of these descending neurons. How are they connecting the brain with different motor areas in the fly's ventral nerve cord? Previously, Jeremy and I, working on this problem, had come up with a kind of circular representation, and we were working to put our information into this circular pattern. And we decided that we perhaps needed to go to a more linear arrangement of the information in order to make it clearer.
What's going to be on this column here is like this circle unrolled.
Right. I think in this meeting we really had a breakthrough about how to capture something that's unique about the information we have, which is really the individual identity of these descending neurons.
What we just came up with was basically reorganizing that circle so that now the inputs are on one side of this chart and the outputs are on the other. And then you have these elements in the middle, each one of which represents a descending neuron, and then the connections all come in to that circle in the middle and then all fan out, which means now we can add levels of interactivity where people can, for example, just explore this by saying, oh, I want to see for this DN, what are all the inputs and what are all the outputs? And by reorganizing things dynamically based on some analysis, we can actually look for patterns in either the input or the output or these elements in the middle.
Often connectome representations can treat entire cells as populations, or you're just looking at connections between regions of the brain. And we really wanted to break that down one level further to represent individual neurons. And so I think our breakthrough was really how to add that level of information to this visualization.
These things take a lot of iteration, especially when you have sort of data that's kind of messy and hard to reason about, hard to represent. It takes many iterations because you don't immediately see the answer. That's sort of why it's hard in the first place, and you have to structure it and compute things and do analysis and look at it lots of different ways. This is basically my office. I do have an office, but I spend most of my time in Bob's. The reason I like working in Bob's is that I get to see everybody over the course of the day. I think it's the way this place is built. Everyone comes by Bob's at some point during the day, and I work with a lot of different people on a lot of different kinds of projects, and in order to keep my mind going, I want to be interacting with all of them as much as I can. So if I'm in Bob's, Gwyneth can just come by for half an hour, and we can talk about something, and I'll probably get an idea for another project.